So there's two flavors of transduction. So we looked at generalized and specialized. So where is my, let me go back to this. So with generalized transduction, I think the main reason they say it's generalized is it's because the host DNA is all chopped up during this process and it's just floating around in bits and pieces. And so you see that in the middle picture. And so any random piece of that host DNA might get packaged into the viral particle. So the gray viruses down on this last image are showing that. So now you have to imagine when this cell lyses open, it will allow for those phages to get out. And if a phage that has some of the host DNA captured in it goes to a second host and injects that into the second host, then that second host may get genes from the first host that it maybe never had before. And it's something that could help them adapt better, give them that adaptive fitness to their current environment. And so the general idea, that word is general because it's, pretty much random pieces of DNA that can get taken up by the virus. And then we compared that to specialized, where we look at the idea of it's a little bit more complicated in the sense that the viral DNA first pastes into the host DNA. And that's what you're seeing at the very top, where you see the phage DNA, the dark green. So that's the viral DNA that pasted in to the host. And then under normal conditions, that viral DNA eventually will get cut out of the host, and then that virus becomes active. And it's usually a clean cut, where it's just the viral DNA that comes out. And then so off to the left, you're seeing what happens under normal conditions. And then the virus becomes active, the virus makes more particles of itself, and then it busts out of the cell. But sometimes you get a situation where the cutting of the viral DNA is not clean and it takes some host DNA with it, flanking host DNA. It's not random. It's flanking pieces of DNA, specific pieces of, depending on where the virus was pasted in. And then you can see that it's taking some of the host DNA and packaging that into the virus. Now, when that virus busts out of this cell, goes to a second host, it may inject successfully that host DNA, the first host, into the second host. And so the second host, if it survives the viral attack, maybe that DNA that came along is going to help that second host. It just happens. Viruses aren't always super, they make mistakes when they go through their replication. Viruses mutate incredibly rapidly, and they make mistakes. So sometimes that happens. And then, sometimes the DNA that they end up taking up is so like maladaptive to the virus that they just don't even survive. They're like, wow, nothing's going to happen. They're not going to make any progress. So, but with this idea of the second bacterial host could benefit from some of these viral particles that accidentally, so to speak, took up some of the flanking DNA with it. Are there questions on generalized versus specialized? Okay, so compare and contrast as you're studying that and thinking about those concepts. I think we left off here where we were looking at the idea that in general, bacteria are kind of cautious about incoming DNA because that often means it's a virus that's trying to take that cell hostage, so to speak. And so those restriction endonucleases that you might work with in intro to cell and molecular, they're coming from bacteria. There's all kinds of them. You can order all kinds of different restriction endonucleases from companies these days. They've discovered that bacteria have a bunch of them out there because, of course, there's a bunch of viruses trying to attack, or phages trying to attack the different bacteria out there. So here they're trying to show the scissors cutting up the incoming DNA. So that's representing the restriction endonucleases. Sometimes the bacteria will adapt by methylating their DNA to try to protect their own DNA from breaking apart during this process. So it's like their immune system. It's like they have a immune system that can help them with this. So what they're finding when they take environmental microbes, not the lab rat microbes, but the ones out in their real life out there, and they look, we say, you know what? It seems like most bacteria especially have viral DNA in their chromosome. And so the fancy way to say that is the first bullet point. Most bacteria in natural settings are lysogens. That's the other. So they're a lysogenized cell. They've been lysogenized. And so you can also say, oh, they're lysogens. That's the type of cell they are. They're lysogens. And that just means when you hear that word, you should immediately think, oh, they've been infected with a virus that stuck their DNA into the host, pasted their DNA into the host. So one advantage to that from the bacterial host is that if you have been lysogenized, you can't get infected by the exact same virus. So if the exact same virus comes along and tries to get into a successful infection pathway, it's not going to work. And so that's definitely an advantage if the bacteria can survive becoming lysogenized and hopefully that virus just stays dormant and never gets active. Then if that exact same virus comes along and tries it again, the bacteria is armed, so to speak, or immune to that second exact virus. So that's definitely a good thing for the host microbe. Did that have that in there? Let me just double check something. No. So I think there's a new term here that I want you to pick up on. So in this image down here, right where I'm circling in red, that cell, and they say prophage, and they're pointing to the viral DNA. So that's a special term, prophage. If someone says to you, oh, I know this cell has a prophage, what they mean is they know that viral DNA is in that host chromosome. And then that stretch of viral DNA is referred to as a prophage. Pro meaning before. I think why they named it that is pro means before. And so it's like before it becomes infectious. So when it's like that, when it's inserted into the host DNA, the viral DNA is inserted into the host DNA, it's quiet. It's not infecting the cell actively. And the cell isn't at immediate risk of death by, true active viral infection. So another thing they're showing here is that when that cell that I circled kicks into, eventually if everything's going okay, that cell might say, you know what, I want to go through cell division. Well, when it does that, what you can see is that part of the cell division process is that cell will duplicate all of its internal contents, including its DNA, which includes the prophage. So now when those two progeny cells are made, the prophage is in each one. So that's a really slick way for a virus to spread into other cells, very passively, very cheaply, very efficient. Just hang there and wait for the bacteria to go through cell division and you're spreading. So that's a really slick way for phages to disseminate or spread out into subsequent, populations of these progeny cells. So that's kind of a sneaky way for viruses to get spread around. So when there is viral DNA pasted in to the host DNA, it can give some unique advantages they've discovered to some bacteria, depending on what the virus is and the stretch of DNA that gets pasted in. So for example, they've discovered that this bacteria, Corynebacterium diphtheriae, which causes diphtheria, this one has to have prophage in it in order to be a pathogen. And so it's, if you've had med micro, this will make sense. It codes for an exotoxin. So it has to have the exotoxin gene, and that's what makes this bacteria pathogenic. So if that viral particle has that exotoxin gene and it gets pasted in, then Corynebacterium can become pathogenic and cause diphtheria. So that means that, you know, theoretically you could work with Corynebacterium diphtheriae, and as long as the viral DNA is not there, that exotoxin gene's not there, you're fine. You're not at risk of getting diphtheria. So there's another example too, Clostridium botulinum, which is really, really seriously deadly. The gene will code for a neurotoxin. And if that viral gene isn't there, then this Clostridium is technically, you could drink a whole test tube of it as long as that prophage isn't there, and you'd be fine. So I used to work with Corynebacterium diphtheriae in this class. And the way I would do that to keep everyone safe is that I would order the A virulent. So right in the, you know, as you go to their website, they'll give you options. And you can buy the virulent one, which means the prophage is there, or you can buy the A virulent. A means without. And so what they've done is they've cured, they call it curing. They cure the cells of the prophage. And there are ways to do that. You essentially force the prophage to excise out, and then I don't know all the details, but they managed to keep the host alive and kill off, you know, destroy the viral part. So you can do that. I stopped doing that because I was, people who aren't microbiologists and don't know that little detail, if they hear I'm working with this Corynebacterium diphtheriae, like the administrators here, they freak. So I stopped doing that. I remember one time when we were interviewing for Dr. Sharp's position, you know, he was interviewing for it too. And we had three microbiologists come to give talks on what their research expertise were, you know, like if they came here, what kind of research would they do. And one was working on the bacteria that causes the plague. And it freaked the administrators out. And I was just kind of giggling to myself because he worked with the avirulent version. He was trying to understand, you know, the bacteria a little bit better. It would have been totally safe. But I mean, I guess if you don't have that microbiology knowledge, I guess that could freak you out. Maybe. So I understood there. He didn't end up getting the job, not because of that, it was other issues. And Dr. Sharp, you know, we won with him. So anyways, if you have that understanding, you know, technically an avirulent Clostridium isn't going to hurt you. So there are some, you know, if you gain those exotoxin genes or the neurotoxin gene, that could make you a very successful pathogen. All right, so that's an advantage for the bacteria as well. So being immune, that's an advantage. Getting access to genes you never had before, if it confers an adaptive fitness to your current environment. Okay, but you have to remember, it's to the current habitat the microbe is dealing with. It could provide a selective advantage, but not always. It depends on if that gene, if they express the gene, if it's really going to help them where they currently live. So now that you have a basic understanding of gene transfer, and I hope that I've got across the point that, you know, this is pretty common. The more they study it, the more microbes they find are doing this. And so that's why they use the terms promiscuous and cosmopolitan, because they know that it's kind of a regular thing. And so you may ask yourself, well, is that happening in me? Because your intestines are full of billions of microbes. And so could this be happening? And of course, the people who are studying the human microbiome, you know, that has been a question that they have been pursuing. And the short answer is yes. They can document that this is happening. And not necessarily in a bad way. I'm not saying it's conferring things that are dangerous to you or, you know, will become dangerous, but they can show that gene transfer is happening. And especially if you start to think a little bit more about your intestinal tract and how well colonized you are, and that there are microbes that are hanging out in close proximity. I mean, you're packed with microbes. And so they're in close proximity, their opportunity to interact with each other is huge. And they're kind of in you know, it's a tight space, so to speak. They're really well packed in there. And so Just because of that, they have more opportunity to do this kind of thing than if they were out in a diluted area where there's not that many other cells around. So they are really thinking about the antibiotic resistance idea too. Is antibiotic resistance spreading in your intestinal tract amongst your microbes? And that is something that they're actively looking at. There's no hard data out there yet in terms of anything causing serious problems, but I think it's something that is of concern and something that you should be aware of. So I'll do, as we get into the human microbes section of the semester, I'm going to do another hunt for whether or not there's been newly published stuff on this idea. Let you know what I find. But, you know, it does have some pretty nice conditions, your intestinal tract. Those are pretty good conditions for the possibility that certainly gene transfer and antibiotic resistance genes get transferred. So having said that, you know, I try to limit how much antibiotics I take, or that my kids were growing up. I tried to limit that quite a bit. Sometimes you need them. Sometimes antibiotics are lifesavers. So sometimes you just have to take them. But when you do take them, the problem is so many of the antibiotics that doctors prescribe are what we call broad spectrum antibiotics. And what that means is they kill off the good microbes in you too, as well as the pathogen, and then that puts you at a disadvantage. So I'll talk more about that when we get into the microbiome of humans. But just this idea of gene transfer, this figure is trying to show the different mechanisms. And I think everything that we've talked about is on here, looking at conjugation, transformation, transduction, intra and interspecies. So they're saying, you know, cells of the same species, but also different species can be sharing. Remember, it's not just one E. coli cell to the other. And so we have a lot of opportunities in the microbiome. So keep that in mind. Then don't forget too about loss of genes, that can happen. And microbes are pretty good at this. They have, it's a survival mechanism for them. They're so tiny. You can imagine that it takes a lot of resources to maintain your DNA and all those genes. So if you don't use that stretch of DNA for a while, it might make a lot of sense to just purge it, get rid of it, because it's a lot of upkeep. And so genome reduction is a thing. It's a very good strategy for many microbes. So for example, Mycobacterium leprae, that genome has been reduced quite a bit, over half. And so they're non-functional or they've been deleted altogether. And so if you are an obligate pathogen, that makes sense. Why hang on to stuff that, you know, if you constantly associate with a host and you always have your host, then it does make sense to kind of purge away what you can rely on your host to do for you, because why keep all that stuff when you have to maintain it if you're getting it for free, so to speak, from the host. And then don't forget about gene duplication. That's another strategy that often doesn't get talked about so much in evolution because it's kind of a microbe thing. Microbes are very good at this. And so they can, you know, they're normally haploid. They just have one gene, one copy, I should say, one copy of each gene. But sometimes when they're replicating themselves, they can do a duplication event. And then what tends to happen is that new gene kind of gets, I like to think of it as being played around with a little bit, changed a little bit, so that it can actually become something totally different than what it started with. And that can actually help. Of course, maybe it'll allow for that microbe to better adapt to its current environment. So duplication is another thing that microbes are pretty good at. Then when they start looking at horizontal gene transfer, kind of, across all the different phyla of life on this planet, they came up with this. One of the studies mapped it out this way. And here they're looking at how many other organisms have DNA that came from bacteria. And the bigger the circle is, means, a bigger percentage of their chromosome came from bacteria. And so you can see that protists, for example, they got a lot of DNA from bacteria. But even primates, we fit into the primate category, and you can see that. But when you start realizing, think about it. The first cell on the planet was some kind of ancient protocell, a bacteria, prokaryotic cell. So it was microbial right from the get-go, 3.5 billion years ago, maybe even older than that. And then, life evolved from that. So, it kind of does make sense that we do have a lot of microbial DNA in us. But a lot of people kind of go, huh? When you talk about that, if you haven't had this kind of background. But, we have a lot of viral DNA in us as well. And they are saying that a lot of these stretches of DNA actually helped us be successful mammals. And so I'll talk a little bit more about that when we get into the human microbe. Okay, so horizontal gene transfer. That's all I have time for this topic. Are there any questions on any of that? Well, I don't know. Let's try an iClicker and see what we can do with that.